Okay, welcome back to the second part of this video on bacterial growth. So hopefully you've had a go at those three questions. Let's go through them together. So I'll switch on the second camera here. Got my calculator at the ready. So the first one, bacteria lands on a cake at the right temperature. How many would there be after 5 hours if they multiply every 20 minutes? So a bacteria, so the first thing we want to know is that it's one bacteria and they multiply. They double every 20 minutes. 5 hours, there's 3 lots of 20 minutes in every hour, times the 5, so that gives us 15 generations. 15 generations in 5 hours. That's 1 times 2 to the 15. And as I showed you before, we switch our calculator on, and that should just go in like this. 1 times 2, use the special button there, to the power 15, close the bracket because I like it neat, and that gives us 32,768. Okay, and that's our first question. Right, next. So we've got 40 bacteria landing on a cake at the right temperature. How many would there be after 4 hours if they multiply every 20 minutes? So we've got, on that one, 40 bacteria landing, 40 times 2, how many generations? So we've got 4 hours, multiply every 20 minutes, so that's 4 times 3 generations an hour, so that's 4, 8, 12. 40 times 2 to the 12, there we go, so we'll stick that in there. So we're starting with 40 times 2, special button there, to the 12 equals 163,840 bacteria. 840 bacteria is the second one. And our last calculation, if 100 bacteria landed on a cake at the right temperature, how many would there be after 6 hours if they multiply every 30 minutes? Now, trick here, we've got a slightly slower growing bacteria because not all bacteria grow at the same rate. So this one is every 30 minutes. Generally, the bigger they are, the more complicated they are, the more DNA they've got in them, the more they have to copy, the slower their generation rate. So this one, who started with 100 bacteria there, we've gone times 2, and how many generations? So we've got 6 hours, 2 generations an hour, 2 lots 30 minutes in every hour, so that is 12 equals, so that's 100 times 2 to the power 12 equals 409,600. So hopefully you can see those are pretty big numbers in terms of the answers to these questions. Okay, they are absolutely enormous great numbers. You end up with an awful lot of bacteria in not a lot of time in ideal conditions. Now, of course, this is an ideal conditions scenario. In reality, bacteria never get these ideal conditions, so they grow always a little bit slower. This is a theoretical maximum. So, moving on then. So, next part of this then, alright, so how can we mess with it? If we know what their ideal conditions are like, then we can obviously slow them down or speed them up as we would like to. So, to prevent bacteria from multiplying, you can either raise or lower the temperature. So if you freeze food, it slows down the enzyme activity inside them, and it slows them down from multiplying, from doing all the metabolic processes and from multiplying. It does not stop them. Freezing does not kill bacteria. Okay, it simply stops them from working. You can freeze a colony of bacteria and then thaw it out and it will come back to life again. And things do go off in the fridge, and they do go off very slowly in the freezer. Okay, you've got boiling and cooking our food, which of course kills the bacteria by denaturing enzymes in them. Do not say, right, kills the enzymes, because enzymes are not allowed. Similarly, denaturing bacteria, no, that's not really what we're after. Kill bacteria, denature enzymes. That's what we want. Prevent them from getting the nutrients. Of course, the reason they don't grow to take over the planet and become a ball bigger than Earth is there isn't enough stuff for them to do so. So eventually they are self-limiting. They'll get to a size where they cannot grow anymore. And then bacterial poisons, of course. So disinfectants, antiseptics, and antibiotics, which we're going to move on to now. So There's a difference between those two words. Disinfectants and antiseptics kill microbes, right? Some of them are safe to use on you. An antiseptic is safe to use on your skin, but a disinfectant probably wouldn't be. Neither of those things are safe to use inside you because they would quite happily kill your cells as well as killing the bacterial cells. So things you might use on a surface or on the outside of the body would be disinfectants and antiseptics. Okay, you certainly wouldn't want to use a disinfectant like Dettol to treat a bacterial illness inside you. That's where antibiotics come in. They kill bacteria inside our bodies because they're not toxic to human cells, but they are toxic to bacterial cells. We'll talk about why in a moment. For example, penicillin, streptomycin. When I say these long words, ampicillin, erythromycin, all these kind of things, people don't seem to know what I'm talking about. Most people at GCSE age, if you go, the banana tasting medicine you had when you were a kid, and then everyone goes, oh yeah, that's penicillin, right? Usually that's how they dress it up to make kids take the stuff. So, how do you prevent them from growing? We've said already, temperature, nutrients, disinfectant, antiseptics, and antibiotics are the main ways we prevent them from growing. So how can we test which ones are the best? Now this is a standard lab experiment and a standard exam question as well. It's basically looking at a plastic plate or glass plate with a stuff called agar jelly in it. Now agar jelly is basically just a jelly-like substance derived from seaweed, which is used to grow bacteria on. You can mix all the nutrients in and it will grow. Okay. Now, if you spread bacteria all over the surface of that agar jelly, then you end up with what's called a bacterial lawn. If you left that alone, it would all grow and become this kind of cloudy here. So this kind of cloudy shows you where bacteria have grown. Okay, so that cloudiness is bacteria having grown. The clear areas here are where bacteria has not grown, where it's been killed. Now, what's happened here is a bunch of little bits of paper soaked with, in this case, different antibiotics, although you could use disinfectants, anything that you suspect might have an effect on killing bacteria, right? You can put them on your plate, right, and you can see how well they work. Whatever you've soaked into the paper will soak into the agar, diffuse out, spread out, and if it's very effective, it'll make a massive hole in the bacterial lawn. So in this one here, this little thing I can just about see says P on it, penicillin. This bacteria not overly susceptible to penicillin, but some of the ones over here, right, TE, Fox, are very, very effective because they've made big gaps, and this one here, E, which I think might be erythromycin, has made a massive, great dent in the bacteria there. Okay, now it's really key at this point that we say that antibiotics do not kill viral pathogens. You cannot take antibiotics for a virus, particularly now at this time in the world. You cannot go around saying that. That's a very dangerous thing to say, because if you take antibiotics for a virus, all you do is kill off the bacteria that your body needs, as discussed in a previous video. Also shown in a previous video was this thing here. So lots of people seem to think they need antibiotics for everything. They do not work for everything. All they do is kill bacteria. So what they do to do that is they actually target parts of the bacteria that you don't have. So there are bits of cell, thinking back to the starter of this lesson, that you don't have that bacteria do have. If you can make a drug that destroys those parts, it'll kill the bacteria while leaving you alone. Okay, so some examples for that might be cell walls. You don't have a cell wall, bacteria do have a cell wall. So if you can put a drug in that stops the formation of that cell wall, you're going to damage and kill the bacteria. That's how penicillin works. Penicillin is part of a bigger family of antibiotics, and that's how they all work. You've got other ones here. So DNA replication has got slightly different enzymes in bacteria to you. So your enzymes that do DNA replication, a bit different to the bacterial ones. Make a drug that targets those replication enzymes, you're going to kill the bacteria, not going to kill you. Same with the ribosomes, which you should know because they're on the GCSE spec, make protein. The ribosomes are a bit different shape and different size to our ones. So you can make a drug that that disrupts the function of ribosomes and therefore kills the bacteria, okay, while leaving you alone, and so on and so forth. Okay, so they only work against bacteria, and sometimes the bacteria mutate and change so that even those antibiotics don't work against those particular bacteria, and that's what we call antibiotic resistance, but that's something for another day. Okay, that's that. Thank you very much. Make sure you've had a go at those questions and you're happy with doing those calculations, and we'll see you next time for vaccinations.